Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on earth, and that's biology. So thank you so much for joining my new beginning session. So here we are talking about biomolecules. So thank you, and please don't forget to like, share and subscribe. So we have started giving you an introduction about what are biomolecules, how did we actually find out, through which experiment, so which part of the biomolecules we are talking about. So we're already done talking about your amino acids. So today we are going to talk a little bit about lipids and then move on to what are nucleotides. Then we're going to talk about what are proteins and polysaccharides, so that will give us a wholesome understanding. So yeah, so that is the agenda for today's lecture. So I hope you guys are going along with me or reading the corresponding NCERT concept, and you guys are solving questions. And if you guys could not watch a live lecture, so make sure you watch the recorded one and get back to me for any doubts, right? So shall we begin? Yes. So here we go, biomolecules part 2. So before I go to today's concept, so I will also revise what I spoke in the previous lecture, that is on Monday. So here we are. So I actually spoke about the chemical analysis experiment where you have to take the homogeneous tissue, where you have to take the living tissue, you have to homogenize it and with trichloroacetic acid, and then you filter it and you get two fractions, right? So that is where we started. So let's start from there itself. So you take a piece, so we are doing the experiment that is called as chemical analysis. Right, so you take a living tissue, so you homogenize it with CCl3, that is trichloroacetic acid, to precipitate the proteins. So once you homogenize, so you get a uniform paste. So now you try to do the filtering. So when you filter with the cheesecloth, you get two fractions. One is acid soluble portion, so this is our filtrate. Then you have the acid insoluble portion, so that is the retentate. So the acid soluble portion has biomolecules which has less number of weight, or the molecular weight is very less, so it is comparatively in less Daltons. Yes ma'am, a very happy good evening. So thank you so much for joining my lecture today. So please don't forget to like, share and subscribe. So this is called as biomolecules or biomicromolecules. So when you talk about the acid insoluble portion, so they are much heavier in weight, so molecular weight, so you can say they range around more than, so this is from 800 to 1000 Dalton, this is more than your 800 to 1000 Dalton. So this is your biomacromolecules. So who comes under micromolecules and macromolecules? So you have your amino acids, you have your lipids, so you have your nucleotides coming under your biomolecules. So here you have your proteins, your polysaccharides, and then your nucleic acids, which is coming under this portion. So I also told you about a small twist in the story, that is your lipids, though it is your biomolecule, it comes in the biomacromolecular fraction, because lipid is always conjugated, like in terms of phospholipid or your glycolipid. So it cannot instantly leave everything and come in this fraction. So your lipids are always obtained in this fraction. Alright. So further we did another experiment. So that is your destructive experiment. To see or to have an evidence that yes, inorganic molecules are also present inside your tissue. So as usual, so you take a living tissue and you dry them. Once you dry, so that is without water, right? That is how when you dry. So after that you burn the tissue. So when you burn, all the carbon containing compounds are volatile, so they evaporate. So all the carbon containing compound or all the organic compounds they are volatile, so they evaporate. Right? So whatever is remaining is your ash, and that ash is a proof of your inorganic components in your tissue. Alright? Yes, dear W, hello, a very, very happy good evening. So thank you for joining my lecture today. So please do like, share and subscribe. So then we started talking about amino acids. So in the last lecture, so amino acids, so you know the structure of amino acids. So you have a center carbon, so that is your alpha carbon. You have a carboxylic acid group, so you have an amino group, you have a hydroxyl group, and then you have your R group. So it is this R group that varies for each amino acid, and you have 20 amino acids totally, right? So if this R group is replaced by H, that is the simplest amino acid, that is glycine. So amino acids can be classified into many ways based on many types of categories. So first of all, we can classify them as essential and non-essential amino acids. So essential amino acids cannot be synthesized in your body, it has to be supplied through your diet. So non-essential ones can be synthesized by your body. So apart from that, you can classify amino acids as hydrophobic, hydrophilic, polar, non-polar, etc. So these are some of the ways of classifying them. Yes sir, a very, very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Alright, so these are the different ways by which you classify. So I hope you guys, yes, what is your basic question? So you still have a doubt and the confusion in the basic concept of biomolecules? Yes W, what is your doubt? So I told you have to remember one positive amino acid, at least one negative amino acid. So positive amino acid, I hope you remember arginine, lysine and histidine. Yes, so negative amino acid can be your aspartic acid, your glutamic acid. So these are all your negative. And we also spoke about your neutral amino acids. So I told you to remember serine, right? So apart from that, biomolecules. Okay, so inside your cell you have macromolecules, right? So when we spoke about evolution, I told you that first you have inorganic molecules. Inorganic molecules became organic molecules, and thus your cell formed, right? So there is some organic molecules. What are those organic molecules? So this organic molecules in your living tissue is called as your biomolecules. So anything that has a carbon, for example your proteins, your polysaccharides, your nucleotides, nucleic acid, they all have a carbon backbone. So anything that has the carbon compound inside your tissue is called as your biomolecules. Yes, okay. So biomolecules is very important because you should know what your living cell is made of. Yes, we know a cell is made of mitochondria, your cytoplasm, your nucleus, etc. But what are the components that drive your cell? So yeah, you know that your house will have a bedroom, a hall, a kitchen, etc. But who's going to live there? Who's going to play a role? So what is going to be synthesized? So that is the important issue. So biomolecules live and rule the cell, right? So inside your cell you have your proteins. Without proteins there is no genetic machinery. So you have your polysaccharides, that is your glucose, your sucrose, your starch, that is how our energy is stored. Then you have your nucleic acid, that is where our genetic information is stored, right? So biomolecules is always given a high priority because when you move into your first year of medicine, so you have a paper called as biochemistry for which this forms the base. So that is why we read biomolecules now with a lot of significance. And yeah, the marks, I would say the weightage is around, so probably it depends. So you can have 3 to 4 questions coming from biomolecules. It is really important. But you're lucky in the sense that you don't have to read the structure of complex proteins and polysaccharides. You just have to know the concept here. But in chemistry, probably I guess you'll be, the same topic is there, biomolecules, where you'll be reading the structure. Yes, understood? Fine. So moving forward, so we also talking back about your amino acids. So we also spoke about something called as a Zwitter ion stage. So your body has various pH starting from your saliva, your stomach, your intestine, etc. So your amino acids have to travel all through the diverse pH, right? Yes. Very good. So I'm happy that you understood, but it's good that you ask me questions like this so that I make your doubts very clear. So unless and until your objective, your aim is very clear about why we have to study, then nothing will enter your head. So very good. So please put forward questions like this. Yes, I will clarify it. All right. Yeah. So your amino acid has to transfer through the diverse pH in your body. So at physiological pH, amino acid has a neutral charge. So this is the neutral, this is your amino acid at a physiological pH. It has both positive charge as well as negative charge. Thus the net charge is equal to 0. This stage of the amino acid is called as the Zwitter ion stage. Right? So sometimes it can be only negative charge. It can have only negative charge at high pH, right? At low pH, it can have positive charge. So this is the Zwitter ion stage of your amino acid, all right? So this is where we stopped about amino acid, and then we went on to talk about your lipids. I'm sorry. Yeah, so talking about your lipids. So lipids are, so I hope you understand the twist. So lipids are low molecular weight compounds. They come under the biomolecular category, but in the experiment they are found in the retentate part because they are always conjugated with something. So please understand, so lipids are actually biomolecules. So if you see here, so lipids are always biomolecules, but they come in the insoluble portion because they are always conjugated with your phosphates, with your carbohydrates, etc. Okay, so that is why it is. High pH, low pH कैसे? See, in your stomach you have low pH. The pH is 1.8 to 2. In your intestine, the pH is pretty high, 6.8, right? So you have varying pH inside your body, inside various, inside the pathway of your alimentary canal itself. So in each case, the amino acid takes up a different form. Okay? Right. So lipids come under the biomolecular portion, but because it's always conjugated, it is found in this fraction. So talking about lipids, so lipids are made up of fatty acids and glycerol. So it is made up of 3 molecules of fatty acid and 1 molecule of glycerol. So when we talk about lipids, the first thing that students ask me is, ma'am, which fat is good? Saturated fatty acid or unsaturated fatty acids? So let's talk about that. So we have something called as saturated fatty acids and unsaturated fatty acids. We also have something called as polyunsaturated fatty acids. Okay, so saturated fatty acids have no carbon-carbon double bond. Okay, so example of your saturated fatty acid is your palmitic acid, your stearic acid, etc. Okay, so unsaturated fatty acid has carbon-carbon double bond. So example will be your oleic acid. So polyunsaturated fatty acid means they have more than one double bond. They have more than one carbon-carbon double bond. Example is your arachidonic acid. Right? Ma'am, this is all fine. Now tell us which is good fat, which is bad fat, and that's a billion-dollar question sometimes when I ask the students. So your unsaturated fatty acids are essential to the body. Your unsaturated fatty acids are essential to the body because your body cannot synthesize them, right? So your body cannot synthesize unsaturated fatty acids. And also, so 3 ke pehle you call it polyunsaturated fatty acid only, there is no specific name. Also, your unsaturated fatty acid remains as liquids in room temperature. One more thing, your unsaturated fatty acid is highly reactive, so it can easily be metabolized. Thus, your unsaturated fatty acid is good for your health. So once again, unsaturated fatty acid is essential because unsaturated fatty acid cannot be synthesized by your body, and they remain as liquids in your room temperature, and they are very reactive, so that is the reason they can be easily metabolized. So it does not get accumulated in your arteries, so it does not create a block. Okay, so whereas saturated fatty acids are exactly the opposite. So saturated fatty acids are non-essential because your body can synthesize them, and they appear as solids in room temperature. They are not reactive, so they get deposited wherever there is space. So it causes fats, it causes obesity. Alright, so with fats, are we clear? So glycerol is known as trihydroxypropane. So you can have monoglycerides, triglycerides and diglycerides. So based on the melting point we spoke, so there are certain oils which remain in liquid in room temperature even in winters, and oils like coconut oil freezes. So why so? So coconut oil has high melting point, whereas gingerly oil has high melting point, and thus it changes its state of matter. Alright, any doubt so far? Clear? Okay, so today we are supposed to talk about nucleotides. So I'll give you a quick recap about nucleotides, and yeah, this is why I want to talk about nitrogenous base and all. So I guess I've already taught this to you when we were talking about your molecular basis of inheritance, but still, so we'll talk about them. So if you see here, so we are going to talk about nucleotides. Okay, so I'll explain you on a blank slide so that it's easy for you. So we are going to talk about nucleotides. So nucleotides make up your nucleic acid. So this nucleotide has two components. One component is your nucleoside. The other component is the phosphate. That is the PO4 minus. Fine? Yes? Yes, what is your doubt, W? Yes, please ask. So nucleoside again is made up of two components. One is your nitrogen base and the other one is your sugar. So the sugar in case of your DNA is your deoxyribose sugar. And in case of RNA, it is your ribose sugar. And here your nitrogenous base is of two types. The first one is your purines, the second one is your pyrimidines. So adenine and guanine are your purines. So here you have your cytosine, uracil and thymine. So cytosine is common for both DNA and RNA. Okay, so uracil is present only in your RNA and thymine is present only in your DNA. So this is the structure of a nucleotide. So see if you guys can understand why it is required in your body. Lipid is very important because without lipid, a lot of things will not happen. Your body needs phospholipids. Again, it's a lipid, otherwise your cell membrane will not be formed. So your cell membrane is phospholipid bilayer. If you do not have lipid, how will that be formed? And your lipids are needed in the process of cholesterol synthesis, and cholesterol is needed in your body. You have good cholesterol, you have bad cholesterol, so good cholesterol is needed. Again, you need lipids. Alright? Yes? Fine. So this is about your nucleotides. So it's made up of two components. One is nucleoside, the other one is phosphate. So your nucleoside is divided into nitrogenous base and sugar in case of DNA is deoxyribose sugar. In case of RNA it is ribose sugar, and nitrogenous base is purines and pyrimidines. So purines are double membrane ring-like structures. So this is your purines. Okay, and pyrimidines are single ring-like structure. So your adenine and guanine is your purine, your cytosine, uracil and thymine are your pyrimidine. So if you look, this is what I was saying. So there's a nitrogenous base and there is a sugar molecule, right? So living organisms have a number of carbon compounds. They are heterocyclic rings. So some of the nitrogenous bases are adenine and guanine. So adenine and guanine are your purines. Cytosine, uracil and thymine are your pyrimidines. So nitrogenous base is attached to the sugar. So when it attached to the sugar, it forms your nucleoside. And when this nucleoside is attached to a phosphate, it is your nucleotide. So your nucleoside will be adenosine, guanosine, thymidine, uridine and cytidine. So this is when your nitrogenous base is attached to the sugar. So this is when this nucleoside is attached to the phosphate, they form your nucleotides. Alright, yes. So DNA and RNA are nucleic acids and they consist of nucleotides only. Fine. So yeah, I told you purines are double ring structures. So you have one ring, another ring, but purines are single ring structures. Clear? So you can see it more clearly here. So again, it needs a lot of nitrogenous base, right? Okay, so I'll finish up what is primary and secondary metabolites so we can move further. So probably when you eat something, so I guess all of you had lunch. So say for example, all of us had a bit of rice. So when you say you had a bit of rice, so rice is starch. So starch is broken down into your glucose molecule, and we have done digestion chapter, right? So we know how digestion happens, how the process of breakdown of food happens. So when you have eaten rice, so the starch in the rice is broken down into glucose. So that product that you get out of a physiological process is your primary metabolite. The process of breaking down food is metabolism. The end product of metabolism is a metabolite. So the food I eat is broken down into a form that is understandable by the body, that is glucose. Right? Now this glucose will be used to make a lot of other things. Say for example, this glucose will be used to produce energy. This glucose will be used to produce my protein. This glucose will be used to produce my unsaturated fatty acid. Right? So this is how it is. Metabolites are the end products of cellular regulatory products. So amino acids, sugars, if you have had chapati, if you have had a bowl of dal, so which means your body has digested it and broken it down into proteins, right? And sugars. So in plants, so now these will enter another pathway. Now this will act as the raw ingredients for another pathway to produce different products XYZ. Those different products XYZ are your secondary metabolites. Alright, so in plant cells, fungal and microbial cells, your alkaloids, your flavonoids, your rubber, your essential oils, your spices, your antibiotics, your pigments, everything is a secondary metabolite. So where your glucose and amino acids are used as substrates to produce a product. Alright, so you have alkaloids in plant, that is a secondary metabolite. The plant synthesizes food, glucose, and this is used to synthesize all these compounds. So these are secondary metabolites. So alkaloids like your morphine and codeine, terpenoids like your monoterpenes and diterpenes, essential oils like your lemongrass oil, toxins like your aberrin and ricin, so lectins like your concanvaline, drugs, windblastin, curcumin, polymeric substances like your rubber, gums and cellulose. Alright, yes, fine. So we'll stop here. So with this we are done with biomolecules. Next we'll be talking about your biomacromolecules. Only two acids are primary. No, amino acid is primary, sugar can be primary, fats can be primary sometimes, fats can be used to synthesize cholesterol. So a lot of primary components are there. Alright, yes. Fine. So we'll stop here and we'll continue on Friday. So please ask me if you have any doubts. So please solve a lot of questions and get back to me on Friday. So Friday 4:30 p.m. lecture 3 of biomolecules. So until then, please have a nice day, nice evening. Take care and bye.